right, so it's glad to be here this morning. I'm Byron Hewitt. I'm co-founder and CEO of Brava Diagnostics, and we're transforming acute care by bringing to market the next-gen test for heart attack for emergency medicine. So, Uncle Jerry winds up having chest pain. What happens? He goes to the ED, and they are going to immediately do some things for him. They're going to take his history, they're going to do an EKG, and they're going to draw blood to get a troponin. Troponin is a protein that's released from heart muscle when it's damaged. So, if he's got a total blockage, he's sent to the cath lab, he's going to get worked up, he's going to get the care he needs. If he's got a partial blockage, he's going to basically going to have to do some more work to figure it out. So high sensitivity troponin provides a faster rule in and a faster rule out. So this is an important advance. So today there's 8 million visits to the ED for chest pain in the United States. About 53% of them are admitted that don't have an MI. So 87% of the time they've not had a heart attack. So the challenge for the ED doc, of course, is to figure out what's going on. So where does troponin come in? We're going to help reduce this excess cost and reduce patient frustration. We can do this because high sensitivity troponin allows you to detect the protein in the blood 4 to 5 hours earlier, which allows the doctor to make a better decision sooner. So what's happened here recently is a study was published in Europe called the Historic Trial. There were 31,000 patients that were part of this study. It was 7 institutions. They basically walked in with chest pain. They then followed this particular protocol, and what they were able to do is with a 99.5% negative predictive value, if somebody had a level of 5 nanograms per liter or lower, they could safely discharge them. This is a huge advance for the ED, no increase in adverse events, and a bunch of these people can be sent home that were previously in the gray zone. So we're excited about this, to be able to bring the first point-of-care assay to the market to do this, and the estimated cost savings in the United States alone is $7 billion annually. So the care team wants it fast, simple, sensitive, and precise. The core lab typically takes 60 to 90 minutes. The reason for this is they have to draw the blood, transport it to the lab, centrifuge it, run it on the system, and get the answer back. And none of the current point-of-care tests meet the new guideline. So Brava is going to be able to deliver this in less than 15 minutes from whole blood using a novel technology that's a planar waveguide. The sample is simply added to this cartridge, inserted to the instrument, and you get your result back in less than 15 minutes. So none of the current point-of-care tests meet the guideline. The contemporary assays that most labs are currently using don't meet the guideline. Only the high-sensitivity assays that are FDA cleared meet the guideline today. So that's Abbott, Roche, Beckman Coulter, and Siemens. So we believe Brava can be the first one to deliver on this promise. And so the market today is about $1.1 billion. It's about half contemporary, 1/3 point of care, and about 20% high sensitivity, but the transition is happening. So we're skating to where the puck is going to be. And so it's not just about sensitive and precise, it's about fast, it's about simple, and it's about the ability to multiplex. Here gives you a little blow up of cartoon of the cartridge. There's a molded lens in here which creates a standing wave of light from a laser that's used to interrogate this. These are where antibodies are placed in a microarray inside the cartridge. This cartoon on the right shows you how it works, but there's a calibrator. You can put antibodies in there, positive and negative controls. And so when we add our sample, the proteins and detector antibodies are going to bind. The standing wave of light means that we're only measuring proteins, the binding events at the surface. We're not seeing other stuff that's unbound in solution. So this is a very elegant and robust approach to a problem that's been around for a long time. And one of the other things about this that's wonderful is that we can use a simple CMOS chip like in the camera in your phone to do the detection, along with this low fluorescent background resin for the plastic cartridge itself. And so this is all patented. We license this in a global license exclusively for Brava in the acute care field, and a bunch of great patents around this. The market opportunity here is interesting because Roche, who's the market leader globally, has no revenue at the point of care in the United States. Neither does Radiometer, who's owned by Beckman Coulter. Abbott gets most of their revenue from the iStat in the US. They have very little ex-US. So it creates an interesting opportunity for disruption for us to bring this to market. There's a bunch of strategics that we believe will be interested in acquiring us downstream, but you get the idea. This is an interesting opportunity that we think they will be quite intrigued by. The management team here has been in the diagnostics industry for 25 plus years each. And so starting out with Abbott early in my career, you can see the team here. Dave has a phenomenal track record of product development in the diagnostics industry, so we're delighted to have him join us. And Carrie, who's here also, has been with me in previous companies as well. We have a fantastic medical advisory board also. So these gentlemen on here, most of whom have done most of the publications in cardiac markers over the last 10 years. So we are thrilled to have them on board. And so the market need is here is defined and understood. We know the market. The technology is proven because it's actually being sold in the veterinary diagnostic and water safety testing markets today. So that reduces our risk from a manufacturing perspective. Our clinical and regulatory development is informed by those that have been through before us. There are no reimbursement issues. You've got a great advisory board, you've got a strong management team, and multiple options for an exit. Questions?
Thank you, Byron. Question from our judges.
So I just want to ask about your IP. It's all around the detection, point-of-care detection, right? Not the assay itself.
That's right. So it's all about the delivery technology itself. So it's the cartridge, the use of the laser, how that laser mates to that cartridge, and all the intellectual property that goes around that. There will be some assay patents as well, but it's on how the assay is done. It's not on the antibodies themselves. They're available commercially.
Do you have any one-to-one testing planned with your product versus the ACEP, for example, and other products that are out there commonly used to do? I know you said the point of care doesn't meet the benchmarks or for whatever the regulatory guidelines are, but they are being used. And they're exclusively being used right now to do this exactly what you're saying it should be done. So I'm just wondering, like, why would you say yours is better? Like, do you have any of those testings planned for, like, to determine that you've been able to catch people who have had MIs better than those? Right. Are you going to be doing that?
Yeah, so it's a great question. For sure, I think what's going to happen is with the market shifting towards high sensitivity troponin, I think that basically the current point of care methods are going to wind up falling short of the mark. Because if they're not sensitive enough, they're not reproducible enough, it's going to be a problem for them. So the idea is if we can be either first or second with this capability, we think it creates an opportunity for a big shift in the market very quickly, because by the time we get to market, it's roughly 2 years from now. And so more of that transition to high sensitivity troponin will have taken place by then. And Abbott's central lab method just got FDA cleared about a month ago. Yes, ma'am.
Thank you. So I just wanted to ask, there is still some resistance to use it. Hello? Can you hear me now? Now can you hear?
I still can't hear.
Hello?
Go ahead, try again.
I'll say it loudly. Okay. Hello?
Yeah,
okay, there we go. So I just want to ask, so how is your marketing for getting past the resistance sometimes to selling to high sensitivity troponin? You're saying you're going to market in about 2 years, correct? And how are you basically trying to sell it to the people who maybe are not using it yet? Do you have a plan for that, or are you really just looking at the people who are already doing it?
Yeah, we think there's an opportunity with this technology to actually move some of the testing from the central lab to the point of care. Because if you can pick up an hour or 45 minutes, and the fact that a third of the market has already voted to do it rapidly, we think, frankly, the reproducibility and sensitivity has not been all that great compared to the current central lab methods anyway. And so it's this tension of speed versus quality. So Frank Peacock, who's one of our advisors, told us in our advisory board meeting, he says, Byron, I can't get what I need today. Today I can get fast and poor quality, or I can get slow and high quality, but neither one actually gives me what I need and want in the ED. So our goal is to deliver that so the ED doc can have what they want. That's the goal.
Is that research you're doing on that as well? Sorry. Is there going to be research on that as well?
Yeah, so when we do our clinical trials, right, so we'll have roughly 12 sites around the country that'll be part of these trials. We will not only do the trials for the FDA submission, but we'll also have user studies so we can address the issue about current point of care versus new point of care, current, let's say new point of care versus current high sensitivity troponin central lab. So we will do those studies so we've got data that supports all that.
Byron, great presentation. One question: can you talk about the new guidelines, the FDA guidelines that you were referring to, and the time frame and when they have to be implemented for high sensitivity testing?
Yeah, so they're not actually the FDA guidelines. They're the American College of Cardiology and the European Society of Cardiology guidelines. And what they came up with is the 4th universal definition of acute myocardial infarction. And what that says is you have to have a 10% CV at the 99th percentile. And so what that means is now what you see with the central lab methods that are FDA cleared, they have a 99th percentile that's down around 15 nanograms per liter. Okay, so this is like the ISTAT is around 100 nanograms per liter. So this is a significant increase in sensitivity and reproducibility from those current point of care tests. And what happens is you do pick up more MI, but you also can rule people out sooner. So it's narrowing that gray zone basically from both sides. And so there's not a timeline for the implementation, but what's going on is the FDA is not going to approve new methods for troponin unless they're high sensitivity, right? So that's where the market's going. And I think that historic study with the 31,000 people in it is it's a landmark study. It's probably one of the best diagnostic studies that's ever been done.
All right, Byron, thank you very much.
Thank you, Mark.
Brava Diagnostics.